So I got this email from a recruiter a few months ago that says, I am working with a small and profitable biotech startup that scaled from 0 to $48 million ARR in 3 years without any VC funding. They've built the fastest DNA sequencing platform on the planet. So of course I immediately knew they were talking about Plasmidsaurus. Robert and I have gotten 3 different variations of something like this from different recruiters at this point. And it stands out because every other recruiter email is like, this mission-driven AI startup has raised $100 million to build the future of intelligent generative voice agents for enterprise. So in short, there really aren't a lot of companies that are profitable this quickly or ever in tech, let alone in biotech, which is even harder. So can you tell us the story about how Plasmidsaurus started?
Yeah, absolutely. It's funny about the thing, like how much they've raised. When we were starting and just starting to recruit, I would talk to people and I'd say, oh yeah, we're making money, excited to grow the team, and they would say, oh, but how much have you raised? And I was like, no, we don't need to raise. We're doing good. And they're like, oh, well, you'll get there. You'll get there was what they would say. So, it's a different way of thinking about things. I started the company as a scientist, as a research scientist who is, I've always been very interested in building a company, which, ultimately means providing value to your customers. And I was working, I was at Caltech as a scientist, and I had my own group, and we were very interested in building, cell therapies that were going to use, synthetic biology. I was working with Michael Elowitz, which is a good synthetic biology lab. And I really wanted to build like the operating system for synthetic biology. That's where my headspace was. And I also feel very strongly that if you're going to do something like that, you have to sort of take a shot on goal yourself and not just build infrastructure that you hope somebody else uses. So in that context, I was planning on building a cell therapy company. And one of the problems with building a therapy company is there's very long time horizons before you know whether or not it's going to hit. There's a lot of technical risk that, it may be like in 5 years you learn that, oh, human biology actually doesn't work this way, right? Like, whoops, you've lost 5 years. And that's sort of in contrast with like a tech company, a standard software company, where really, most things are achievable with enough resources and cleverness and good people. In biotech, that might not be the case, and you learn that too late. So I was planning on, or what I was actively doing, was trying to de-risk things as much as possible within Caltech so that when we left, I wasn't going to learn, 5 years later that I should have just done this early experiment to learn what was going on. So in that context, I was generating, hundreds of plasmids a week, and so a plasmid is, as a biologist, it's a very easy way for you to get DNA into a cell, and that's important because, DNA and RNA is sort of the language of biology. And if you want to interface with biology, that's the most straightforward and programmable way to do it, is by encoding, all of your stuff into DNA. So we're making like hundreds of plasmids a week, and this is like it's a standard workflow that sort of every biologist does, is generating plasmids, but it's still like pretty tedious. And so there's a lot of steps in it. You have to, design what you want. You need to synthesize that DNA, which typically is through a third party. You need to get it into the lab and then clone it into the vector, the plasmid that you're working with. And that's like the step of cutting it apart, inserting it, putting it into E. coli bacteria, extracting it back out of the E. coli bacteria, and then determining if the sequence of the thing that you stuck in there is what you intended, because it's pretty lossy. It could be, depending on what you're trying to do, it might be only half of the things that you're trying to make work. And so that means you need to do a lot of sequence validation to make sure that the thing that you stuck into the plasmid is actually correct. And so we built out this entire workflow for scaling that up, and the bottleneck for us was DNA sequencing, and that final validation step. And that's pretty depressing as a biologist, I would say, because you see stories all the time about advancements in DNA sequencing, and the cost of a genome is going through the floor, and everything's getting better. But as a day-to-day bench lab scientist, you didn't really feel that at all. So if you're going to interface with the advances that were made in DNA sequencing, that was really like a capstone on a big project, but not a thing that you're doing every day. But the technology was there, and I knew the technology was there. So because we were bottlenecked on what's called Sanger sequencing, which is sort of the traditional way that a bench lab scientist would be sequencing their data, I knew there were a number of other platforms that could provide better data, which would be like Illumina is inexpensive, a little bit slower. There's Oxford Nanopore, which is what we ultimately ended up using, which is fast but was known for having low sequence quality. And I tried both of those methods as an alternative to Sanger sequencing, and the Oxford Nanopore technology worked very well. And it was like a step change in how we were able to do experiments once we got that working. And that, I sensed there was a lot of value in that, to myself. And so then I asked my colleagues at Caltech to send me their stuff to make sure that the technique I was using would work on everything, because you get worried that if you start a business to do this, everybody's going to just send you the impossible stuff, and that's like not a fun way to start your journey. So got that, and that all worked really well. And so then I knew there was a business opportunity there, and sort of quickly started offering that as a service. And because this was my life, is knowing all these bottlenecks, it was very easy for me to understand exactly what the product we needed to make for the customer was without needing to do a lot of customer journey or anything like that. And then really within a couple of months, we had launched the company out of my garage, and we're just from day one having pretty good success. And at the time, Twitter was still a thing that was still useful for science, and it was actually a great resource. And we got a lot of viral spreading through that, which I don't know if it's possible now on any platform. I don't think there is that central hub where people are talking about things.
What made you go viral on Twitter?
The biology tool space has been neglected for so long. It's generally owned by very large companies that are very driven on incremental customer capture, but not really thinking about what's going to be transformative for the scientists, just because that's how these companies are structured. When we launched our product, which involved using nanopore sequencing initially to sequence things you previously would have done with Sanger sequencing, it was such a step change for people that I think they felt like, wow, like life is different now. I can do things I couldn't do previously in a way that you just wouldn't see otherwise. And the reason for this is, if you're a Sanger sequencing company, and there was lots of Sanger sequencing companies across the world, what you were really doing is buying a Sanger instrument from like Applied Biosystems, and then you would buy a LIMS system so that when a customer places an order, it just puts it into some database for you. You would get their DNA and just put it in the machine functionally, and then it would just email the people the output of that machine. And none of that infrastructure existed for us, and so we needed to write all of the core algorithms ourselves for, how do you generate the raw data coming off of the instrument into something that is familiar to a scientist. So we put a lot of effort into that. We had to generate what the actual consensus sequence was for what they'd given us. That's the core thing they wanted. But usually a scientist at the time were used to looking at Sanger traces, and there's quality information that they're familiar with in the form of like an AB1 file. And we had to artificially create those that would surface the same quality issues, but in a way that these were not machines that would generate AB1 files, had to synthetically generate those. And integrated plasmid maps. So previously, if you were using this, you'd get just a string, fundamentally, of a DNA sequence, and you need to figure out what that encoded and use a third-party viewer to generate where that fell on your plasmid map. Well, we would just surface the entire annotated plasmid map right back to the customer, which again just like saved them an enormous amount of time. They also didn't need to generate primers, which is the thing you need to do for Sanger sequencing. So I think it was the thing that made it go so viral was that scientists, the first time they used it, felt like their life had been meaningfully improved. And that's just not really a thing you see elsewhere.
I was a biology major, and I did do undergraduate research. And I do actually remember once sending something off for what must have been Sanger sequencing. And all I remember is that I put something in an epitube in an envelope and put it in a Dropbox or something. Can you clarify what I must have done before reaching that point? Let's assume that we were sequencing a plasmid.
Yeah. So you have a plasmid, which you've extracted out of some E. coli at some point, or gotten from a friend who'd done that. And then you would cut it apart with restriction enzymes. And then you would modify it in some way, which is either like a point mutation or most likely inserting the thing that you care about. Maybe it's GFP or a CRISPR protein or whatever it is. And there was this delusion that the only sequence you need to worry about having been modified in a negative way is the thing that you had inserted yourself and right at that scar. And the reason I think for this delusion was that it was just such a pain in the butt to sequence the whole thing that this is what people had focused on. So to sequence that small part of the plasmid, what you would do is you would first, you'd need to open it in a plasmid viewer to see the region that you've modified. You then need to design primers, which are single-stranded DNA oligonucleotides you could get maybe, say, 800 to 1,000 bases at a time for a single Sanger sequencing reaction. And that's if things go well. If things go wrong, it could be maybe only 500. But you would design your primer just, say 20 bases upstream of where you want that good sequencing read to begin. And then maybe 700 bases later, you would design another one and another one until you got across that entire region that you modified. So that requires designing the primers, which is also, you got to figure out the melting temperature and a number of things about that. Then you need to order those from a third party, like IDT, wait for those to arrive, and then you would include those in this envelope that you would give to the Sanger company. And then they would mix those together when they arrived, put it on their machine, and then they would give you a series of 800 base pair strings that you would then have to align back to the thing you did and try and figure out, did I sequence the thing you want? And so, if you were doing whole plasmid sequencing with Plasmidsaurus, you don't need to do any of that primer design. You don't need to figure out what region you care about. But a nice side effect of this was that people started getting the entire plasmid sequence every time. And what they learned is that, there had been this delusion that the only thing that matters is the part you modified. But there was a lot of parts in plasmids that would go bad over time anyway. And part of this, the reason people thought you didn't have to sequence the whole plasmid is because the E. coli genome is very stable and the mutation rate per base is very low, that you wouldn't expect to see it happening. But these plasmids are not pieces of the E. coli genome. These are things that are expressed at high copy in the E. coli, and so there's selective pressure there to mutate them away. And they also tend to have highly repetitive regions. So if you want to have an inducible gene, say you want to make a system where you can add a drug and a gene will turn on. Well, you do that by engineering a plasmid, and you do it by putting a whole bunch of binding sites, maybe 30 binding sites for some specific protein on that plasmid. And that's a thing that doesn't really happen in the E. coli genome, and that it's not good at replicating. So what would typically happen, and it's also very difficult to sequence that. So what would happen is somebody would once make this 30 binding site motif in their plasmid. They would do a lot of work with restriction digest and all sorts of things to verify that it was what they thought it was, and then never sequence it again. And nobody would want to. Or if it had high GC content, or if it had hairpins in it, all these reasons, you wouldn't really ever go back and resequence it. But all of those are the things that are very structurally unstable, and so they would mutate a lot. And so what we heard from our customers once they started using us was that, this project they were working on had been mothballed for a year after tons of work, and they couldn't figure it. Experiment was going really good, and then it just stopped working one day. And when they sent us the plasmid to try and figure out what had gone wrong, it turned out that this repetitive promoter that they'd created had collapsed down to a single repeat and therefore was no longer inducible, right? Or that the hairpins that were structural elements for something they're doing, had mutated away and been deleted out of the plasmid, or that there'd been other structural rearrangements. And that was really invisible unless you were doing whole plasmid sequencing. So it's really when we first came out, we got emails all the time from people that were like, hey, this experiment had stopped working. In the worst case, there was a PhD student who had spent years trying to figure out why this thing wasn't working like full time. And then she sent it to us and immediately was like, oh, this plasmid was wrong, a bunch of stuff got deleted. But you're never taught that, or you hadn't been taught that at the time, that this was actually even a possibility.
They didn't even know they had to worry about it.
No.
And were people never doing whole plasmid sequencing at all?
Yeah, so essentially no. Essentially you would never do that. You might do a restriction digest to make sure things were roughly the right size. But there's also information, even if it looks the right size, because of the way restriction digest works, if the plasmid had duplicated, which is a thing that we now know is quite common, when you restriction digest, those would collapse down into a single band and become invisible. But if you did, for some reason, need to do whole plasmid sequencing, which is a thing you might do if you were going to go in clinical or something like that before you start generating something for humans, you would do something called primer walking, where you would make a primer, you would sequence 800 bases, look at that region, design another primer near the end of that, and sort of just do that all the way around the plasmid, and then you would know where things are. But that still, we learned, missed some structural variants that you can pick up with whole plasmid sequencing.
So have you had to educate people about this problem?
I think so. We're very much like. We like to think of ourselves as empowering scientists to do the research they want, and it seems they get it pretty quick, right? Once they get that thing back, and they see that in most cases, I would say in the majority of cases, if a plasmid had not been whole plasmid sequenced previously, there's probably a number of things about it that are not the same as what they had expected. So the first time you sequence it, I think you're like, oh, this is a problem.
Interesting. Are there cases where people bought plasmids from a third-party company and those turned out weird too, or?
Yeah, so that's another case. There are third parties who will functionally do your plasmid cloning for you. They will synthesize the DNA and clone it. And yes, we heard a number of cases where those were wrong. One case is people try and use it, and they're like, oh, this is a plant vector, and I did not order this, and it's not used in our lab, and it had just been cross-contamination in the factory from somebody else's plasmid that could come. That's not super common, but does happen. But a case that stands out to me, there was a researcher at Harvard who was working on lentiviruses, and lentiviruses are they're gene therapy vectors that the payload, the part you care about, is flanked by some inverted hairpins, which makes them somewhat structurally unstable. And they're sort of known to be unstable, so you got to treat them a little bit gingerly. They'd ordered this from one of the big names that does this for you, had gotten back Sanger sequencing data with it, had gotten back Illumina data with it as part of their protocol, had gone to use it, and the plasmid didn't work. And they couldn't figure out why it didn't work. And they sent it to us after spending a month or so trying to debug it. And it turned out that it had just been a complex structural rearrangement that is not detectable with those other methods, because everything sort of exists in the plasmid, and at each of the junctions that you would care about is right, but there's higher level structural changes that were not detectable. So they sent to us, and immediately we're like, okay, we need to reorder this.
Interesting. So did the Illumina not detect that because
it's short? So what happens is if you're trying to stitch together what a DNA sequence looks like, if you have repeat stretches that are longer than your read length, it becomes sort of an impossible problem of trying to figure out what goes on the two sides of those repeats, because there's nothing in common that allows you to stitch it to. So that was the case here. And so yeah, each read, you could map it to the vector in a way you wanted, but ultimately there were these high-level structural problems at the repeats.
When I heard about what Plasmidsaurus was doing, of just use Oxford nanopore sequencing, so you have long reads and you can sequence the whole thing, I was like, oh yeah, sure, of course, that seems obvious to me because I've been working with long reads for human genome assembly and structural variant detection and things like that. I guess I have been away from the wet lab so long that I wouldn't have known that people were still using Sanger sequencing, or that there was an opportunity there to actually replace it.
Yeah. I think one of the lucky things for me is I was still doing a fair amount of bench work with my team, and all of the pain points were very obvious to me, and just saying like, this isn't right. Like, there's something wrong about the fact that we don't have access to these tools. And I had previously used Oxford Nanopore for a different project and was familiar with the strengths. And so to me, it was very obvious how to do this and get it out to people. But again, did other people think about doing this? I assume they did, but at some point you have to actually, get out there and drive the change that you want to see. And I think that's, fortunate for us that we did that.
Yeah, definitely. One of the things we find so refreshing about Plasmidsaurus is just seeing a company that actually noticed a problem and then started solving it, rather than kind of the hammer looking for a nail, where these startups like have some technology, they're trying to figure out which segment of the market to target with it, and they're pivoting and they're looking for product market fit and all that. And it's just like really refreshing to see somebody actually being like, oh, here's a problem, we have an obvious solution to it, and we're just gonna start rolling it out.
Yeah, and I would say there's like, if you're working in a research lab, there's a lot of problems. Like sequencing, DNA sequencing is not the only problem, right? There are so many problems that require you to if you come into the lab, to learn some very specific skill set that maybe nobody else in your lab has. And you have to learn that. Maybe you're going to go over to a collaborator and learn it, or just try and learn it from the literature and go through, I would say, like a very, very painful experience of learning how to do it. And still, it's probably, inferior in some way. Some other researcher has published it and a thing they thought would be interesting, but not really focused on simplicity and ease of use and all those things. And I think there's just an enormous opportunity for people to provide services that will offload that work and allow people to get the answers they want. Because ultimately, as a scientist, you have a scientific question in mind. You you have some hypothesis about it may go this way, it may go that way, and you know what experiments you can do that will help you cleave off one of those hypotheses. And that's what you care about, right? Is like actually learning that question as quick as possible. Learning how to become like very, very good at some skill set requires you to sort of like pipette at an angle and not let it dry out too long or whatever. Like that's not really value creating for anybody. So as we can find ways to eliminate that, I think it's positive for biology as a whole.
Yeah. And I feel like sometimes people in the lab have too high of a bar of what they're willing to go through. Like I used to work at Invitae, we did genetic testing, thousands of samples sequenced every day, and every time I would visit the lab just to like shadow what they were doing, there were all these like backflips they had to go through with the software. And they were okay with it because like they kind of got used to it, but there's a lot of low-hanging fruits there, I feel like. But you just have to be in the right mindset to be looking for them.
Yeah, 100%. So a thing that's very important to us, because this is, what Robert's saying here is extremely common, and there's these sort of like gaps between different parts in the workflow, where it could be that if something was just done a little bit different on the wet lab side, it would make the analysis way easier, or alternatively, there's an analysis that seems obvious, that would eliminate a bunch of work that has to happen on the wet lab side. But there's not that communication about like the pain that somebody is feeling every day, and so it sort of requires somebody to really be in the lab and from a software side to be in the lab and understand, oh, this is a problem they're having that's easy to solve, and the problem is like as a scientist, you don't know necessarily what's an easy software problem and what's a hard software problem, and so you're not really suggesting like, oh, could you just do this? Because it might be a thing where, oh yeah, super easy, or it might be a thing like, well, that's going to require like a couple PhDs worth of work to figure out. So, a big thing for us, and we're here at our headquarters in South San Francisco, is very much embedding our bioinformatics team, our software team, our robotics team, and our R&D team all in the same space, with desks together, and having people, sit next to each other when they're doing the work and really understand like, hey, wait, why are you clicking on that 10 times, right? Like why did you go through this weird context menu and open this thing up and do it that way? And they're like, oh, could you make an like typically what will happen is they'll say, oh, if you could make a way to automatically click on that and then click on this and click on that and click on this, that'd make my life a lot easier. And it's like, well, we don't need to do any of that, right? We can just, it's just a single line change here in the code and it'll make your life so much easier. So that's, I think, when we think about the velocity that we are able to move as a company, a lot of it really comes from the fact that our team is just so tightly integrated and understands each other's pain points and what's easy and what's hard. That's very important for us.
It's really interesting because it kind of flies in the face of the advice to like specialize, like you just focus on your thing, you get really good at that, and then let the other people focus on the thing they're really good at. And bioinformaticians might think that the wet lab is repetitive and standardizable, and they don't know if there are things that they could ask for to be done differently.
Yeah, I think that's true, and I think there's always this this sense about automation. So if you're doing a PhD in biology, you're going to spend a lot of time doing repetitive stuff or sitting there and pipetting, from one 384 well plate to another in a way that's very error prone and takes a lot of time and is actually mentally pretty stressful, because even though it's very repetitive, if you lose your focus, all of a sudden you got to start over. And so I think I would say maybe most wet lab PhDs are oh, I just need to automate this, right? If I could just automate this, then that's my big idea, is I'm gonna automate this. The problem with that is that automation works really well for standardized protocols that you're doing over and over. A lot of what you're doing as somebody working on a PhD is you're trying something once. You're doing one experiment, and then maybe you're going to repeat it once or twice. But actually the lift to get that working well in an automated system is probably not worth the cost. And so you do just have to suffer through it for a bit. But that's in contrast to a company, which is why I was saying earlier, there's a lot of low-hanging fruit where companies could do things that speed up what people are doing and decrease the learning curve. Because if you're a company and that's all you're doing, it makes a lot of sense for you to invest in the automation to do that. The other thing that has been, I would say, surprising to me building this company is, as a scientist, you'd always get pitch solutions of a box. Buy this box and it's going to solve this problem for you, right? It's gonna clone your DNA for you, or it's going to, do whatever these things are. And I think the sort of limit of that was when Oxford Nanopore came out with their MinION sequencer, which was functionally free when they released it. You just had to buy the flow cells, and it was just plugged into a USB drive, and it couldn't be easier or couldn't be more accessible to use. And so that's the limit of oh, we just got to get a DNA sequencer in every lab and then everybody can be sequencing all the time. Well, one of the ways we were able to grow so quickly is that people didn't want to do it, right? They would rather have, even though they could have it in their own lab, they would rather not have to learn how to use the thing and worry about why is it erroring and what's the maintenance on it and how do I deal with the data and all those things. It's people don't want to do that. They want to offload as much of the standardizable stuff as possible, and that's pretty obvious where I think if you look at where the industry is going, that's generally the case. But it becomes very difficult when you're working on your PhD and you've got some strange system that nobody else has used and you've come up with it. I think for the time being, you just have to suffer as a wet lab biologist.
Yeah, I love the box thing. An analogy I just thought of as you were saying that is it's like saying everyone needs a bread machine in their kitchen.
Exactly.
And eventually your entire counter is just filled with machines for every possible thing, but I think most of us prefer to just be able to go buy bread at the grocery store once a week instead of having this giant machine sitting on our countertop.
Yeah, no, that's exactly right. You might want to make bread every once in a while. And if you want to make bread, that's great. You can do it, and you can enjoy making the bread, and maybe you want to make a bread company, and you're going to sit there and make bread all the time because you love it so much. That seems great, right? The most likely scenario is you want to make a sandwich, and you don't want to spend a day making bread, even though it's so easy for you to, if you just planned better, you could have dumped in the flour on this machine that's sitting on your desk. It's going to cost you about the same anyway, so why not just get the bread from this?
Right. even if you need a sandwich every day, that's still...
Yeah, even if you've got the bread machine taking up space that you could otherwise be using, you're still probably just going to go to the store and buy a loaf of bread.
Right. So you mentioned there's a lot of low-hanging fruits still out there.
Yes.
Just asking for a friend, can you name some of those for us?
I think, obviously I've got things that are obvious to me. I think realistically, if you talk to your friends who are in, very productive labs and you just sit with them and see how they're spending their day, it's pretty apparent that there's things all over the place. So another product that we do here is RNA-seq, which we can talk about. That's the thing where if you ask somebody why aren't you doing it, they'd say it's too complicated or whatever, or it's too much work. It would be difficult to get for somebody to articulate how much of their research is blocked because there's a bunch of upfront learning on just knowing how to do the technique. And so this is a thing where, and it's like any technique you do, you have to learn how to do it at some point. And there are many things where, for instance, just doing CRISPR screens, okay, which is a thing that's very common right now throughout biotech. But if you've never done a CRISPR screen and you show up in a lab and it's we need to do a CRISPR screen, well, there's a lot of learning you have to do that might take you a couple months of sort of getting up to speed and figuring out the right way to do it, and what reagents should I be using, how should I design my library, how should I do all these things. And then at some point, you're gonna be okay with it because it's a thing you know how to do. And you might not say oh, learning how to do a CRISPR screen is such a bottleneck because you were able to do it. And the reality is it set your research program back by 3 months as you were trying to get up to speed on that. And so if I was starting a company in the space, that's what I'd be trying to think right now, or what are the things where people would really like to be able to do these techniques, and they're maybe fairly common techniques, but certainly not everybody knows how to do them. You could probably start a business doing those things right away, and get customers right away doing that stuff.
I think that does get at the point Robert made before, that wet lab scientists are just used to a lot of pain, and so they don't even complain about it.
It's a good point. Wet lab scientists are used to a lot of pain, and that is actually important for having a productive workflow, I think, as you do have to go through the pain, even if you're ultimately going to automate it. But that leads to a situation where lab scientists also tend to not really value their time as much as they should. And so they might see oh, this is cost $100. Well, I could just do that myself. Why would I pay $100 to do that? And it's yeah, but you're spending all day, right? So there's the cost of your time, which you're undervaluing, which is going to be more than that. And then there's also this opportunity cost of now you're set back more time because you had to go through and do all these things that you could have been reading a paper or thinking about your experiment more. So it's a core problem that scientists don't value their time as much as they should.
Yeah, we run into this exact problem.
I mean, that was exactly the thing with Circa, right? It takes a week to figure it out, or you could pay.
Right, we have software that makes it really easy for people to make sucrose plots, which is something many people have told us takes them a few days of pain on the computer to deal with in R or with the sucrose package itself. And yet they don't want to spend $100 or $200 to solve that problem, even though it's now pleasant to do it too. They're oh, whatever, it just takes them a few days.
It's a bit of a catch-22 in that the very well-funded customers are typically aligned on that. They don't want to wait, they're potentially well-funded because their research program goes very fast, right? And then they can get their papers out and then get their grants. And that means that then they can invest that money in going quick and scooping the competition or whatever the currency is there. And so that matters a lot, and they're generally willing to pay the money to do that. And a less well-funded lab might be getting a lot of direction from the top that, hey, you really shouldn't be spending money on these sorts of things. But that creates sort of a terrible feedback loop, and where now because they're not spending the money, they're not making the progress, and then ultimately you're not publishing the papers and not getting the grants. So.
Yeah, they're in a hard position.
They're in a hard position. They're in a hard position. So everybody's in a hard position right now.
Yeah, they are.
Yeah, so other than being profitable, one thing I really like about Plasmidsaurus is, at its core, it's a lot of logistics.
Yeah.
And that sounds like a lot of fun.
It is fun if you think about it right.
And I have a fun story to share. So when I was in grad school around 2014, I somehow ran a small business on the side where we were printing a bunch of stuff, including name tags for conferences. Super random, but the idea was there were people who, organized a lot of events, and they would send us a list of attendees. We'd print the name tags, put them in the plastic holder, give them a lanyard, ship it. It would be ready for them to go. And as the number of samples grew, number of name tags grew, I guess, in this case, a lot of the logistics got a lot more interesting, where like one time our printing partner swapped the UBS labels and the conferences opened the box and were like, this doesn't look right. And so we had to like tweak the logistics to like, in this case, put a picture of what the badge on top would look like so that when you're closing the box, you could see like color and design differences. That was super fun, on top of being extra stressful, because my PIs didn't exactly know I was doing that. Hey guys. But yeah, like, do you have fun stories like this where you're like tweaking things in the logistics to like make it work better and more like less error prone?
Yeah, absolutely. In logistics, I would say, you describing building a small business, printing name tags, which, you said it's fun. I think most people, if you said, hey, I'm going to start a business to print name tags, they wouldn't think like, oh yeah, it's my life's calling. But I believe you that it's fun because, you were able to do things that people got value from, and that's very fun. And then the other thing is, and maybe sometimes I think you can lose sight of this when you're in a big research program, is like, a mission, it's conquered by tackling a bunch of small problems, right? And each time maybe much like writing code or whatever, and each time you sort of achieve one of those things, it's very gratifying to be like, oh, there was this problem where the things were getting swapped or whatever, and like there's a solution to it, and now it's solved, right? That's a great feeling. We feel the same way as we are doing logistics. So our logistics system is set up that we have over 1,000 of these hyperlocal drop boxes, which are embedded at the institute where the researchers are. We then need to find some way to get samples from those drop boxes into our processing facilities. And we have 10 processing facilities around the world that are sort of strategically located to, really for logistics reasons, to be able to get things into those labs as quick as possible so that, if people want to do plasmid sequencing, they will get their data by the time they get into work the next morning. That's really how we think about it. And yeah, it requires literally, like, we have open labs in different regions so that just for logistics reasons. We have a lab in London, we have a lab in Germany, we have a lab in Singapore, really just so we can get things into those locations as quickly as possible. When we started this, which is analogous to your story, it was us doing everything ourselves, right? So our first drop boxes were at Caltech, and it was like clock out of my day job, start going around the university, picking up the samples, putting them in a bag, and putting them in my trunk, and taking them to our lab. And then it was, okay, now we need to expand beyond Caltech, so go to UCLA and do the same thing. And then it's get off work, pick the samples up from Caltech, drive to UCLA, pick samples up from UCLA, drive back home. And then that's like not a super fun drive. That's maybe an hour each way or something like that. And then, so then that was going well, but it was also like, okay, we need to find an actual more scalable solution for this. So the next place we wanted to go was San Diego. And we knew there was a lot of interest down there, but we really only wanted to make a service that was going to get people data by the next morning. So we hired a courier. We used like an app, an Uber-style app, to find a courier who would every day, pick samples up from around UCSD and the institutes down there, drive them up to Pasadena, and then have us process them, and they would get in at 8 p.m. or something like that. And we did that every day, and that was going well. And then we just the driver who we were working with decided he just wanted to work with us. So he started working with us, and he actually still works for us. Oh. And then we were thinking about opening a lab in the Bay Area, and which is like opening another lab is a lot of work, but we knew that there was a lot of people who could use our service up here. And then sort of just by thinking about it, we realized we didn't actually need to open a lab in the Bay Area. We just worked with couriers in San Francisco, or in the Bay Area, to drive around to Berkeley, to UCSF, to Stanford, to the different labs. And then half of them would go to the Oakland airport, half of them would go to the San Jose airport, put the samples onto an airplane, drop them off there. Those would fly into Burbank. We would have a courier go to Burbank, pick them up from there, drive them into our lab, and then we could process those, right? Which is quite logistically complex, and the airlines would change their flights all the time at the last minute, which would, it was very complex. It was every day, it was like a logistical process to try and figure out how we were getting the samples into the lab that day. I remember there was like a day we missed the flight because there was like a carjacking on the Bay Bridge or something like that, and so like we didn't make it to the airport in time, and that meant that we couldn't fly the samples into Burbank Airport, so we had to fly them into LAX because Burbank was closed at that point, which meant we had to then have a courier that went to LAX at like midnight, picked up the samples, gone back to our lab at like 2 a.m., and then we were in the lab at 2 a.m. because we had to get the stuff on the sequencer so people would get their data back by the time they got it in the morning. And then as you're doing this, you're building out scalable solutions, right? So then you start saying, okay, maybe I can work with third-party contractors to help in some of these locations, or maybe I need to hire people in some locations. You work with, there's FedEx, there's UPS, there's these ones where if we need to get something, from a place where there is not a large demand, how can we get samples into our lab as fast as possible? And then, we have full-time employees who are just thinking all the time about where is each sample. How do we have visibility into where each sample is? How do we alert customers if there's a delay? Yeah, that's a giant part of our organization, is optimizing that in all ways. We have to think about, when there was an announcement, the politics affects this. So there was an announcement about things coming into the US where now there's going to be different rules about what can get into the US. And that was a whole process where we had to spend, a month of just trying to figure out, how can we get things that need to come into the US in there? Do we need to reroute things into Germany, into the UK, to ensure this all works out well? So yeah, it's a lot of work, but I think our customers really feel that they know their things are going to be sequenced as fast as humanly possible, and when there are any sort of delay, we go way out of our way to make sure that they don't experience it.
That's awesome.
So are there places in the world where it takes a lot longer than till the next morning?
Yeah, so Asia is difficult. We do have our lab in Singapore, which is faster than shipping things to the United States. But like Australia, we have a lot of business in Australia. We would love to have a lab in Australia to make that go faster. But the problem is that just, Australia is a lot of cities around the rim of the country, and trying to get samples from one part of the country to another it still is going to require shipping it regardless. And if we're going to do that, we find it's about as easy just to send it to Singapore, and they still get their data much faster than anybody else would give it to them.
Interesting. So some of the videos on your YouTube channel, there are dinosaurs on bikes delivering packages. Would that not work in that case?
We occasionally, we do have bike couriers in a number of locations.
Oh, for real?
Yeah, there are bike couriers. That was when we started. So there's a video that does show bike couriers, and I think that was in Oxford, and that was highlighting that we were using bike couriers there. They don't typically wear dinosaur costumes, okay, although maybe occasionally for fun we do it. But no, I mean we will do anything and everything to ensure that the process is as fast as possible. So it is bike courier. I mean, there's a scenario where you can have a bike courier take something, drop it off at the train, and then a train courier will take it into London and deliver packages that way. We'll do whatever is most efficient for the customers.
Nice. Do you have a dashboard showing where?
Yeah, internally we do. And there's a fantastic software engineer here whose entire job is to make sure that tooling is as good as possible for us to use.
We're just like, we love logistics. It's just so fun to like think about how to make a system more efficient.
So logistics is a thing, like many of the stuff we do, where we feel like we're building durable value by building out this network, right? So now that we have this incredibly strong logistics network, it means we can add additional products and have it be very low friction for the customers. This is similar to a thing we do on robotics. So I think we'll do a tour later of some of the robotics we're working on. But we focus very heavily on what are unique capabilities that we can lever up that will allow us to do things that are otherwise impossible. And it could be that we develop sort of a new way of treating samples for one protocol, but we know that that's going to work across many new protocols. And an example where this recently came up is we launched an RNA-seq product about 2 months ago, and that's been going very well. And we can process a lot of samples at scale, and that's because when we think about launching a new service, it is very much focused on like, how is this going to be as scalable as possible? How can we launch this in all of our labs and not require insane amount of training for the technicians, but more of just like, lay out the robot in this way, push start, and have it go. And that requires like a bunch of custom 3D printing. Obviously magnets are a thing that's used in DNA purification. So embedding those in the 3D prints, actuating things mechanically within the robotics. But as we build that out, then it makes it super easy for us to use those in other things. So for RNA-seq, which is this product we just launched, before we launched our product, you would expect to wait 4 to 6 weeks to get data back from a service provider. And the process was, you negotiate a contract with them about like how much you should pay, and like depending on how much they can charge you and how good you are at pushing back is how much you're going to pay. And then once you get all the PO signed and everything, then you'll get your samples, you put them on dry ice. You will probably do the extraction yourself, or you're going to pay them a bunch of money to do the extraction. You might do the library prep yourself, or you'll pay them to do the library prep. Then they would run them through their DNA sequencer, and then typically you would get back the FASTQ files, which are like the sequence files that you get off of the sequencer. And then either you would pay them to do additional bioinformatics analysis, which is going to be another delay, or you're going to work internally with a bioinformatician on site to do that. And then finally you can sort of learn like, did my experiment work or not? And you're really talking maybe like 8 weeks from when you first thought about doing it to when you have some useful data, and a lot of money because you're being charged for each step. Because we are thinking about things with speed, and we have this logistics network, and we have this robotics infrastructure, and we have a very strong bioinformatics team, and we have a very good software front-end development team, every decision we made when we thought we were going to do RNA-seq was around how can we make this as streamlined and scalable as possible, with the idea that a customer would give us as crude of a sample as possible. So in this case, it's typically tissue culture cells shipped at room temperature. And then what they will get back from us is the answer to their question, which is generally going to be what genes and pathways are different between my treated cells and my untreated cells, or on my time course, how are those things changing? And so whereas the standard method, it might cost you $200 a sample for the extraction and the library prep and the bioinformatics, and may take 8 weeks for you to get your data back. Well, for us, for academic customers, it's only $50, and they're going to get their data back in 3 days, fully analyzed. And when you're thinking about like the life cycle of an experiment that a scientist is doing, typically they are, you might want to think about things on like a 1 week or a 2 week cadence of, I'm going to design a new construct, I'm going to treat my cells, I'm going to harvest them, I'm going to do these things, and then you're going to make a decision about like, is this working or not within, maybe hopefully a week. And that's just not possible if you want to do RNA-seq and you're not using us. Whereas if you use us, it's really about as fast as if you were doing qPCR, which is a much less rich analysis, and it still requires a lot of work for the customer. And so that's the thing where we leveraged our logistics network, we leveraged our robotics network, we leveraged everything we have to be able to create like an entirely differentiated experience for the customers, which is another thing where, again, people are messaging all their friends, I can't believe this is real. And the product growth we've seen from it has been remarkable.
Wow. Yeah, a few questions on that. Are there people who were doing the RNA-seq in-house, and what would that take for them to do it? Or...
Sure. Yeah, and I should say, like, if your life is, running RNA-seq experiments at scale, and there are some core labs that are like this, or there may be like a company or something where this is a lot of what they do. The actual like end-to-end turnaround time that you can get if you want to go all in on it is about on par as how we do things. But that's almost never the situation you find yourself in, because you need to wait to fill up the machine with, you want to fill up all the lanes so that you're not wasting money. Whereas we sort of just by nature of running it so much can run all of the lanes every day, and we don't have to worry about that. The sample prep, again, is typically pretty laborious for a bench scientist. Even if you're at a facility that's doing a lot of this, it's still very manual. Whereas, we had to think from first principles, how can we do RNA extraction and library prep in a way that scales is roboticized, that steps are optimized to not, if it says a 2-hour incubation, we're very sure that that's required, or we would do it faster. All of those steps.
In terms of the price point, how are you able to make it so cheap? And what do you say to people who are skeptical that the quality is going to be lower?
Yeah, it's interesting. So what we would like is somebody to use it every time they're doing a tissue culture experiment. We would like somebody to think like, I would like to know everything that's going on in my cells, as opposed to, I did a Western blot and I see that this band is darker than this band. You want to know, or I did a I think a lot of what's replacing is qPCR, which is where you use quantitative reverse transcriptase PCR to convert the RNA into cDNA. You then put it on a real-time PCR machine, and you can sort of get the quantitation between your samples. That requires having reference genes, GAPDH or whatever the things are that you want to go against, which are sort of arbitrarily chosen.
And is that one gene at a time that you're...
Yeah, so with qPCR, you're going to find a panel of, you say you're interested in some pathway, you're going to find a couple genes in that pathway, and you're going to do those in parallel. It was like, at what price point is it somebody going to say, I would rather just do the RNA-seq on this, get every single gene, than go through the pain of doing this myself and getting only a couple of genes. And we felt $50 was like roughly that point where it would sort of be a no-brainer that you would just send us everything that you would have otherwise done with qPCR. And then like on back testing for that, we found that actually it's more sensitive than cyberine-based qPCR. So when we did a side-by-side, we treated a bunch of cells with interferon, which is like causes known transcriptional changes as a positive control. We did the RNA-seq on them, and then we also did qPCR. And we chose the genes for qPCR based on the literature, not based on our RNA-seq data. And in most cases, they matched up like perfectly, right? So if you look at the plots you would get from qPCR, you look at the plots that we got from RNA-seq, at least like they were very similar. In some cases, the order of magnitude was a bit different, which we believe was due to inefficiencies in the amplification of the qPCR product. But there were a couple of cases where there's big outliers, where it showed like for the control experiment on qPCR showed like much higher expression than we saw on RNA-seq. And in all of those cases, from that panel of like 24 genes that we looked at, the high signal that we were seeing in qPCR was right around the noise limit for the assay. And so what it really was just non-specific amplification. And so like the data is like even more actionable than if you're doing qPCR. And we've heard from our customers, as like I spend a lot of time just going to universities and meeting with people, that people are doing this. They're finding there's a no-brainer that you would just do RNA-seq instead of qPCR.
Interesting. So it's like similar to what you did with plasmids. You wanted to make it like a routine thing that you do instead of Sanger, and this is like another routine thing.
Yeah, and I think similarly, like so with plasmids, what happened was people, they learned, oh my gosh, there's all this other stuff going on my backbone that I wasn't aware of. With RNA-seq, what they're learning is like, oh, there's other pathways that are being activated here that I didn't appreciate. Or actually, there's only a subset of these things are changing when I thought they would all be changing. An interesting example of that was we had a customer who was, she was interested in she had her cells that were, treated and untreated, And as part of her perturbation, she had covered her cells in a thin layer of gel so that they would be protected from the environment, because she was really interested in how the environment was affecting the cells. And she didn't see the changes she thought she was going to see, and couldn't really explain it. So she sent us the RNA-seq cells. And what she learned when she looked at the data is actually the cells that had been covered in the gel were all hypoxic. And so like that's the thing, by looking at qPCR and all these things, you would have never discovered that. You know, her pathway, she was interesting. Yeah. But then it was very easy for her to say, oh, why is hypoxia upregulated in all of these cells?
That would make sense.
But that could have been like a 6-month troubleshooting detour trying to figure out why things are not behaving the way they want, as opposed to it's literally analyzed like functionally. You go to the page the minute you get an email saying your results are ready, and it's like hypoxia is the thing that's changed.
I heard in another interview you said that you set the $15 price point for whole plasmid sequencing so low because you just wanted people to do it more often. You wanted to drive biotech forward.
Yeah.
And it sounds like with RNA-seq, you want people to do RNA-seq more often.
I think they should do it every time they're doing tissue culture experiment. I think like seriously, the amount of projects that right now in this country, in this world, that are stalled because something that could be easily resolved by just knowing what the transcriptomic data is enormous. There are many, many people who are spending months trying to find some cute way to debug their problem when they could just very quickly learn, oh, these are the things that are actually changed.
That's fascinating. So how did you come up with the idea to do RNA-seq?
Yeah, it's, I mean, again, thinking back to my life as a wet lab biologist, a lot of what you're doing is you are trying to figure out what the expression differences are between your treatment and your control group. And I mean, I knew RNA-seq is a good tool, and it's been around for like 15 years or something like that. And there had just been, you know, like we were saying earlier, there just never been a strong effort to say, how can we do this, make this accessible at scale? Because the companies that previously were doing it were very focused on hitting their next quarter's, you know, profit metrics, as opposed to saying like, hey, we can invest, you know, a couple, you know, almost a year in really building this up at scale, really building through a process that is going to work for everybody. Because it just doesn't fit with the business models, I think, that other people.
Can you say more about those other business models, just to help people who don't, help us understand more what's going on in the biotech sector?
Companies that are sort of like commodity DNA sequencing services. And what you're really getting with that is they have some Illumina machines, maybe a couple other instruments, which are capital expensive, like $1 million machines. And you could either write your grant and try and buy this machine for $1 million, or you could pay some third party to use their machine. And their margins are not great, and they every quarter need to say, you know, how profitable we are and how an idea like, so if you're a public company, one of the things you need to be doing is you need to be showing that your profit margins are getting better every quarter. So you're spending less for every dollar that you get in. And the way you can do those things is you can be slow, right? So that you're never having any waste on your machines, for instance. Or you can, not take projects that like small, you can't take a sample. A lot of the people who are using RNA-seq for us, they're sending us 6 samples, right? And they'll just drop it off today and they'll get their data back in 2 or 3 days. For like a company that is really just focused on making as much profit as possible, well, just the customer service of getting them a quote is going to make it hard for them to make money. So it's just, it's not been a thing they focused on.
And like speaking of other, I guess, competitors, so I was Googling Plasmidsaurus, plasmid sequencing, and as usual, the top result was sponsored from another company. Overnight, $15, full plasmid sequencing. I'm like, wait, that's like your tagline basically. Yeah. How, what's your strategy there for?
Yeah, that was, so when we first started, I was very, because we were operating in my garage, and there were a number of these sort of legacy Sanger sequencing companies, and I very much focus on speed of execution, not on IP, right? So if you're like a drug company, you make all of your money by getting patents and then having a monopoly on those and being able to extract as much money as possible from customers because there's no alternative. If you're a tech company, you really win by just out-executing everybody else and getting things to market as quickly as possible and doing it better than everyone else. And that's, from my perspective, that's like much better for everybody if that's how you're doing it, because it just ends up in a better customer experience for the scientists themselves. So that's how I designed everything. But I was nervous that, these big companies were going to wake up to what I was doing and try and copy me, and it seemed like they must have tons of people and tons of resources to deploy on these. So as soon as I started, I was focused on getting to other, as I was talking about earlier, how can we get into San Francisco as quick as possible? How can we open a lab in Boston? How can we do all that stuff? Because I wanted to have all of that stuff at scale by the time these legacy providers started copying us, because it was obvious it's going to happen. And I didn't want them to have a speed advantage on us, which is what they would have if we had to be shipping stuff across the country and they could do it locally. So that was like very early on, made me very nervous. And then it was obvious at some point they would start copying us. And it wasn't clear what the end game of that would be. In the meantime, we focused so heavily on quality and customer experience that I knew that if people were happy with us, and ideally we have tools, and we do have we'll give people back integrated alignment tools so they can see their data aligned with what they would want right in browser without needing to pay thousands of dollars to a company for that tooling. We spent a lot of effort building all of that out so that when these people started trying to compete with us, we would know that we had a superior product. And then they come out functionally the same price. They say the same turnaround time, but that's, I think, not always the case. I know it's not always the case. There's a lot more failures that need to be rerun and stuff like that, or just didn't want to fill it up that night. And the customer experience is just so much worse. So a thing that we focus on very, very intentionally here, and you can talk to our customers and you'll hear it, is we focus heavily on customer support, technical support, really helping people solve their problems. And we had customers at the beginning that would say, I am just it's such a pleasure to work with you guys compared to this competitor they'd sort of been forced to work with, that they're thrilled. And what we found is by just having the best quality, having the best speed, having the best customer support, having the best user experience, there isn't even if somebody is going to pay a bunch of money to Google to try and sneak above us on the search, I don't think it's fooling anyone. And we hear from our customers that they're not going anywhere.
Yeah, and we see it all over Reddit and social media too, that basically the vast majority of comments are extremely positive.
Yeah, that's I would say that's very encouraging when we see those things. It makes you feel great. So if you guys are using a product from anybody and you love it, you should let them know, because it feels great.
Yeah. So comment down below if you like this podcast. Cool.
So what does sequence everything mean? Where are you going after RNA-seq?
Yeah. So sequence everything sort of intentionally has two meanings, right? One meaning is we should be able to do any kind of DNA sequencing, or any kind of sequencing, RNA sequencing, DNA sequencing, protein sequencing once it becomes a real thing. So in that sense, we would like to be able to do any technique that will be enabling for a lot of customers. But the other thing is you should be sequencing every time you do an experiment, right? You should be, no matter what you're working on, the information that would answer your question in many cases is in the sample. You just need to sequence it. And historically, it's been just such a pain to get that information, or it's been financially cost prohibitive, or requires too much work. That for us, what we aspire to do is to make it so easy that you can sequence everything you're working on, and allow you to make decisions as quickly as possible.
That's amazing. I do think that's driving biotech forward, which is exactly your goal.
We hear it, and that makes us feel great, because that's exactly what we're trying to do. But you can imagine, any application that currently is limited by speed or access to sequencing is something we're certainly very interested in. And again, just when we think about products that we offer, we really want it to be, you give us a tube that is ideally as crude as possible. We want to be able to take it without you needing to do any work. You drop it off at your hyperlocal drop box. We get into our lab as quickly as possible. We process it in the way that will surface the answers that you need, and that we have taken, offlifted a lot of that standard bioinformatics analysis so that you can learn, the answer that you need directly on our website. And we do have a very good, pleasant user experience on our website. And I think that helps out everybody by being able to make those quick decisions. And then, I know there's some bioinformaticians who watch this. Like, a thing we really want to be able to do is offload the sort of standard grunt work so that, you can work closely with a scientist on the bioinformatics applications that are specific to their project, because everybody's got their own experiment that's unique in its own way, and it really does help to have a bioinformatician working with you on that, but isn't really necessary for just the boilerplate stuff.
Amazing. So how long until we can spit in a tube and just send that to you?
Let's go to the lab.
All right, let's go to the lab.